Okay. Good morning, everyone. I think it's time to start our session, our conference, and for start, I would like to welcome everyone, speakers and delegates to this conference. On behalf of organizing committee, Fourth International Conference and Exhibition, Cell and Gene Therapy. This conference was initiated in constant encouragement of Journal of Stem Cell Research and Therapy, Journal of Cell Science and Therapy, Journal of Genetic Syndromes and Gene Therapy, and of course Scientific Committee of Omics International Group. And this morning I would like to first of all thank you organizing committee for their tireless efforts to bring all us together to this nice venue and exchange our ideas and maybe start some collaborations. And just to start this session, I have a few slides on gene therapy, some retrospective since mid-80s to 2007 and to dates. Yeah, and basically what I would like to show that how that research was distributed in geographically and in research in different types of diseases. First of all, as you can see, that most of the gene therapy efforts were concentrated on, of course, cancer research, and this data was basically published in the Journal of Gene Medicine in 2007, and since then it didn't change much. And as you can see that still, even though it's shifted a little bit to some other targets such as DMD or Ebola, but cancer research still in kind of center of gene therapy research. And last slide, I would just this is second to last, that how clinical trials progress with evolving gene therapy technologies from antisense era to RNAi era. And as you can see, by 2007 antisense was slowing down, especially when ISIS shut down their large antisense program, focused more on small molecule research. But RNAi therapy wasn't that kind of evolve in large scope of clinical trials and research. And as you can see, by 2007, clinical trials were cut almost in half. But as 7 years later, this slide just updated recently. I did it a couple of months ago with all clinical trials on RNAi. Usually it's short RNAs or siRNA prodrugs and drug candidates. as you can see that mostly with leadership of L. Nylam and Quark, Takeda came with clinical trial 1, and now they moved to clinical trial 2 with Ebola treatments. But if you will follow latest blogs and events on gene therapy, since 2001, when Thomas Tuchel published first a paper in Nature about siRNA in collaboration with Planck Institute, there were no any drugs FDA approved RNAi related. It's kind of discouraging, but if you will see that even antisense technology was more mature in this sense, it took more than a decade and a half to develop first drug Vitravin by ISIS, and then a few months ago, second one was basically FDA approved. It's Kinamra for treatment hypercholesterolemia. That is quite encouraging fact and good perspective for gene therapy area. And at this conference, we will see many reports on antisense study and RNAi-related therapy, as well as cell therapy. So on this note, I would like to invite our first keynote speaker, James Karapatnik. He's director of cancer research at West Ontario University, and Lawson Health Research Institute. So please welcome Dr. James Karapatnik.